students, today we will study about the structure of DNA and RNA. These both are the nucleic acids. And what are nucleic acids? Nucleic acids are the biomolecules. Biomolecules means these are the molecules that are found inside the body. And these are found in the cell nuclei and in the form of nucleoproteins. It means this nucleic acid is combined with a protein, histone protein, and this is called the nucleoproteins. So firstly we see the full form of DNA and RNA. DNA, the D stands for deoxyribo, N for nucleic and A for acid. So DNA is deoxyribonucleic acid, while RNA, R stands for ribo, N for nucleic and A for acid. So RNA means ribonucleic acids. Actually these both are the polynucleotides. What is polynucleotides? The word poly means polymer and it is the polymer of nucleotides. It means the nucleotides are present as the monomer units. It means several nucleotides repeats again and again and they form a polymer structure, that is polynucleotides. So DNA and RNA both are the polynucleotides. If we see the nucleotides, the components of the nucleotides, if we hydrolyze it, then we get know that the nucleotides consist of three basic units. These are a sugar, that is a pentose sugar, a base, that is a nitrogenous base, and the phosphoric acid. Firstly, this sugar and the base combines. It means one sugar and one base combine and forms nucleoside. And then this nucleoside combines with the phosphoric acid and forms nucleotide, that are the unit of the DNA and RNA. So nucleotides are basically the phosphoric acid esters of the nucleosides. Firstly, we see the structure of the sugar. The sugar are of two types. These are 2-deoxyribose sugar and the ribose sugar. Actually, both of these are the pentose sugar. It means they possess 5 carbon atoms. The 2-deoxyribose sugar is found in DNA, while the ribose sugar is present in RNA. This is the structure of deoxyribose sugar. This is a pentose sugar. These are 5 carbon atoms, 1, 2, 3, 4 and 5th carbon atom. And in this case, the second group, in this second carbon atom, the hydroxyl group is replaced by this hydrogen atom. There is 2 hydrogen atoms, no hydroxyl group. So this sugar is known as 2-deoxyribose sugar, and this sugar is present in the case of DNA. While in the case of RNA, ribose sugar is present. This is also a pentose sugar and both they have the same structure, almost same structure, but in this case at the second carbon atom there is a hydroxyl group present. So it is a ribose sugar. If we see the configuration of both the sugars have the beta configuration. It means the hydroxyl group is present at the upside. This at the first carbon atom, this is upward, so it is beta configuration. We see the structure of the bases that are present in DNA and RNA. There are 2 types of bases. One is purine and another is pyrimidine. Purine bases, their structure resembles to the purine. It means they possess the purine skeleton. So these are called purine bases, and these are basically 2 types. Adenine, that is represented by A, and guanine, that is represented by G. The pyrimidine bases are 3. Cytosine, that is represented by C, uracil, that is represented by U, and thymine, that is represented by T, out of which the adenine, guanine and cytosine are common in both DNA and RNA, and these are found compulsorily. But uracil is found especially in the case of RNA, while in the DNA, uracil is not found. Thymine is present in the DNA. So these 2 are optional. This is found in the RNA and this is found in the DNA. So in total, RNA and DNA both possess four type of bases. In DNA, the bases are adenine, guanine, cytosine and thymine. While in the RNA, the bases are adenine, guanine, cytosine and uracil. If we look out the structure of adenine, this is the structure of adenine and this possesses the purine base at which this amino group is substituted. So it is a purine base. And guanine, this is the structure of guanine. It's also possesses the purine skeleton and in this there is a amino group and this keto group is also present in the case of guanine. Structure of the pyrimidine bases. These are three: cytosine, thymine and uracil. Firstly, see the structure of cytosine. This is the structure of cytosine in which there is a amino group and a keto group present at this pyrimidine skeleton. This is the pyrimidine skeleton. While in the case of thymine, this is the structure of thymine and there is a methyl group and this is the total skeleton of it. One dissimilarity between uracil and thymine is that in this case of this is the structure of uracil. In this there is no methyl group present, but in the thymine there is a methyl group. Both have almost similar structure, but it differentiates between the presence of methyl group in the thymine, and this thymine is found in the DNA, while in the RNA uracil is present. Now we see the structure of the nucleoside. What is nucleoside? It is the combination of sugar with the base. It means when the sugar combines with the base, then a nucleoside is formed. Suppose this is a base, that is adenine, and then this combines with the sugar, that is deoxyribose sugar, and it combines at the C1 carbon atom of the deoxyribose sugar, and as a result of this, that combination is formed. This is called the nucleoside and it is deoxyriboadenosine. This is the adenosine nucleoside, and in this way four different type of nucleosides are formed because there are four different bases. So every nucleic acid possesses four different nucleosides. If we look at the structure of the nucleotide, what is nucleotide? It is the combination of the nucleoside with the phosphoric acid. It means when a nucleoside combines with the phosphoric acid, then this unit is known as the nucleotides. Suppose this is a nucleotide. In this is the nucleoside. We have studied that this base combines with the sugar and forms the nucleoside. Now this nucleoside at the C5 carbon atom of the sugar, this phosphoric acid group get attached. As a result of this complete unit will be known as the nucleotide. So in a nucleotide there is a base and a sugar and a phosphoric acid group, and this base is attached at the C1 carbon atom of the sugar, while this phosphate group is attached at the C5 carbon atom of the sugar. So this is especially the adenosine phosphate, and there are different four bases. So there are four different nucleotides and their particular arrangement gives a specific character to the nucleic acid. Now in the dinucleotide, it means when two groups get attached, two monomer units get attached. How will they get attached? At first carbon atom, the base is attached. At the fifth carbon atom, this phosphate group is attached. Now this third carbon atom also possesses the hydroxyl group, and this hydroxyl group is substituted and this phosphoric acid group of the another nucleotide get attached at this. It means two nucleotides get linked by this phosphoric acid group and this dinucleotide is formed. We see the structure of DNA. According to Watson and Crick, the DNA possesses double helical structure. It means it is double helix. It is comprised of two chains that are coiled around each other. that is called double helical structure, and each strand of it is comprised of a polynucleotide chain. And if we see the backbone of this chain, this is the backbone of the chain, and this is made up of sugar and the phosphate group. If we see at the molecular level, it is formed of phosphorus, oxygen and carbon. Now, the hydrophilic phosphate groups are directed outwardly while the bases are directed inward side. These phosphate groups are directed outward and these bases are directed inward. It means towards the center of the molecule. These both chains are anti-parallel. It means the C3 end of the one chain and the C5 chain of the another chain are near to each other. It means one chain goes upward and the another chain goes downward. And these are the bases that are joined by the hydrogen bonds, the two strands, and the nitrogen bases of the two strands are joined to each other by the hydrogen bonds, and this forms the bridge like structure, like the ladder like structure between these two strands. Each turn is in the length is of the 34 angstrom. The length of each turn is 34 angstrom and there are 10 base pairs in between one turn. It means the gap between two bases is 3.4 angstrom. And if we see the diameter of this molecule, the diameter of this molecule is about 20 angstrom. If we see the molecular structure of the DNA, this is the molecular structure of the DNA and it comprises of two strands. These are the two strands and each strand is comprised of a polynucleotide chain. If we see this is the one nucleotide. This is one nucleotide and several nucleotides are joined together. In each nucleotide, this is a sugar, that is deoxyribose sugar, and base is connected at the C1 carbon atom of this. This is the base that is connected at the C1 carbon atom of the sugar and at the C5 carbon atom. This is the C5 carbon atom. A phosphate group is attached. As this is the C5, so this end is known as the C5 end of the DNA. Now at the C3 carbon atom, there is attached the phosphate group of the another ribose sugar, another nucleotide, that is forming a dinucleotide structure. It means the phosphate group is attached to one C5 carbon atom and at the another end to the C3 carbon atom. In this way a link is formed. This phosphate group, this is attached at the C3 carbon atom here and here at the C5 carbon atom of another nucleotide. In this way a chain is forming. This is called a polynucleotide chain. Now you can see these phosphate groups are directed outward side. These are hydrophilic. It means they are attracted toward water, and these are directed outward sides. And these bases that are hydrophobic in nature, it means they are repelled by water, and these are directed inward. It means toward the center of the DNA molecule. So this is the C5 end of the strand and this is the C3 end of the strand. Now if you see this another strand that is anti-parallel, it means the C3 end of this is in this side and the C5 end is in the another side. So it looks like inverted structure. You can see this is inverted because this is C3 end and this is C5 end. So we can say both the strands are anti-parallel to each other. Now the bases are directed inward sides. So they can be bonded to each other by the hydrogen bonds. And this provides a ladder-like structure between these two strands. These bases have a unique code. It means their arrangement is very unique. And this provides a special code to every DNA molecule, and that is unique for each individual. Now, when we see the hydrogen bonding between the bases, there is a unique type of hydrogen bonding between the bases. It means a special base binds with a special another base. That is, the purine base binds with the pyrimidine bases. It means thymine will make the bond, hydrogen bond with the adenine, and it makes two hydrogen bonds with the adenine, while the cytosine makes three hydrogen bonds with the guanine. This is the structure of thymine and this is the structure of adenine. As you can see, this is the oxygen and this is hydrogen, and there is a hydrogen bond between them, while this hydrogen makes a hydrogen bond with this nitrogen, because a hydrogen bond can be formed with the electronegative atom and the hydrogen. Now if you see cytosine, this is cytosine and this is the guanine. Now there are forming three hydrogen bonds. One hydrogen bond is this, another this, and the third one is this. So in this case three hydrogen bonds are formed, while in this case only two hydrogen bonds are formed. DNA possesses the equimolar amount of the guanine and cytosine and the equimolar amount of adenine and thymine. It means the amount of guanine is equal to that of cytosine, while the amount of adenine is equal to that of thymine. Why? Because there is a special type of bonding, that is hydrogen bonding, between guanine and cytosine and between adenine and thymine, and this further proves that the DNA possesses the double helical structure. Structure of the RNA. The RNA is the single stranded. It means it is consist of only one single strand, and this strand, the backbone of this strand, is consist of phosphate groups and the sugars, while these are the bases, and here the bases are adenine, guanine, cytosine and uracil. But here the amount of guanine is not equal to cytosine and the amount of adenine is not equal to uracil. Why this is? Because there is no double helical structure and no bonding, no hydrogen bonding between the bases. So their amount may be any. It is not definite that the amount of guanine will be equal to cytosine or the amount of adenine will be equal to uracil. They may be any number. And the RNA are basically of three types. These are the mRNA or messenger RNA, tRNA or transfer RNA, and the rRNA or ribosomal RNA, and these are involved in the protein synthesis. So this is all about the structure of DNA and RNA. If you like this video, please share, please comment. Thank you.